So I want to welcome everybody to this evening's talk. Tonight we've got Kerri Frangioso, who's going to be our speaker. Kerri has studied the ecological impacts of sudden oak death in the Big Sur region for over 20 years. We were just discussing the fact that she started working on one of her field sites on the preserve back at the very early days of the preserve in 2020 or 2000 or 2001. And has been a partner and friend of the Conservancy for a very long time. She's a staff researcher for the David Rizzo Plant Pathology Lab at UC Davis and has spent countless hours, probably months of her life in the field observing and documenting wildfire and the interactions with disease such as sudden oak death. She has lived up in Big Sur since 2001. She's up at the top of Colorado Canyon with her husband and a menagerie of animals, as she says. And she's got all kinds of experience with fire, both research personally and as a very active community member. And we've worked on some fire policy stuff together, which has been a real pleasure. So without further ado, she's going to share some of her updates on her research.
Well, thanks, Christy. I really appreciate it. And it is true that I've been working with the preserve staff for many years. In general, the working here on the preserve has been one of the easiest places I've ever had to work with and do research on. And I think that what the preserve has is very special. I talk about it often with people. So I think what you have here is really great. And a lot of great things happen here that don't happen elsewhere. So yeah, I have been studying sudden oak death for over 2 decades now. And so I've seen a lot of changes. We've learned a lot of things, but there's still a lot of things that we don't know. And so whenever anybody asks me to give a talk, it's hard because I could talk for a long time about lots of different things about sudden oak death, but I'm going to just give you a brief overview of what I am going to talk about. For people who don't know about sudden oak death, I'm going to give a background about what Phytophthora ramworm is, what causes sudden oak death, and then we're going to talk a little bit about management opportunities, and then I am going to kind of transition into I've done more on the research front and looking at the interactions between wildfire and sudden oak death, specifically here in the Big Sur region, and then briefly touch on our future challenges and the direction of our research right now. So back in the summer of 2000, I was hired to go and examine an unknown forest pathogen that was killing tan oaks by the hundreds, mainly in the Bay Area and in Sonoma and in the Big Sur region. So this picture is taken in Big Sur, lots of dead tan oaks at that time. And there are a lot of reports coming into the different agencies, so many that they could no longer ignore it. And so later that year, the causal agent was identified, Phytophthora ramworm. So Phytophthora literally means plant destroyer. And a lot of people refer to Phytophthora ramworm as a fungus, but in actuality, it's not a fungus. It's an oomycete, which is a water mold and more closely related to brown algae in the ocean than it is to mushrooms. And it behaves kind of like a fungus, except for that the spores that it releases when it is spreading actually have swimming structures that can propel it through water. So one of the really important things with Phytophthora ramworm is that it needs water to go through its life cycle. It's very, very much water dependent. So here you can see the sporangia on the left, and those are the actual swimming structures. And then in the bottom right, those chlamydia spores basically form a shell around the organism, and so that when the conditions are really bad, it can basically overwinter. Or here in California, it would be over summer, where it's really, really dry and there isn't water, so it can basically survive a dormant period. How did it get here? Well, we know that it was introduced, unfortunately, through the nursery trade, unbeknownst to all the people in the plant trade, and specifically on rhododendrons. And what happens is that in the nursery trade, they often are treating their plants with fungicides and herbicides so that when you go and look at your plant, it's going to be beautiful. It's not going to have any bad looking leaves or anything. And so, and that makes sense. And so what happens is that if it had some sort of a disease, you wouldn't know it because you bring it home and you plant it and it would look beautiful. And then as the fungicides would wear off, it would then be able to spread and cause disease on the plant that you had. And given the right environment and given the right host, it can then spread out into the surrounding forest. And that's exactly what it did with Phytophthora Worm, causing sudden oak death in California. One thing that as consumers that we can do, and this is one great thing that we can do, is that if you're unsure when you go to a nursery whether or not these are disease-free plants, because if there were any diseases on them, they would likely be suppressed by fungicides, is to self-quarantine your plant. So if you purchase a plant, you can then set it aside in a hospitable location for the plant, water it and keep it alive, but let it sit there for a couple of weeks before you plant it. And at that time, the fungicides would be able to wear off. And if it had any disease, you would then be able to start to see symptoms, and you could bring it back to the nursery and say, hey, look, I've got this disease plant, and they should be very concerned about that. And likely give you a refund. But that's one thing that you can do, is self-quarantine your plants. And unfortunately, this problem of introduction of pathogens through the nursery trade is ongoing. It's an extremely difficult challenge around the world. And it continues to happen to this day, even though people are trying very, very hard to not introduce new pathogens into the world. One thing, it took us many, many years to actually find out where Phytophthora worm is from. If you can imagine, this is an organism that doesn't have a fossil record. Nobody really cares about these organisms until they're actually causing problems. So there's not really a baseline, there's no baseline data to say, okay, these are our native Phytophthora in this location. And so it took us over 2 decades to actually find a location where you could find both mating types and you could find genetic diversity in one location. And that location was in along the Vietnam and Chinese border at a high elevation forest that have a lot of the same plants that we do, not the actual plants, but the same plants from the same genera. So they have oaks and they have laurelaceae, which are related to bay laurel, and they have beech trees, which are affected in Europe, which I'll talk about a little bit later. And so it's really interesting that the organism has developed these pathways to affect the same kind of plants that we have here. But these plants, because they have grown up with it and they are used to being able to identify it in its environment, they don't get sick like our plants do, because our plants have no idea what this thing is. They have not evolved with it like these plants have. So they found lots of different Phytophthora worm, and they have 8 different lineages and different isolates. So this is pretty new. And so we hope to learn a lot from this information. How do these species of plants actually live with it? And is there a way that we could learn from how these plants can live with it and incorporate that into some of our plants here? So looking at the genetics of the pathogen, you can see that it was introduced into California about 8 different times before it caused an epidemic. What's really interesting about this is when you look at the genetics is that it's essentially a clone of itself. So I was just talking about over in the Vietnam-China border, there was genetic diversity and there were both mating types. So there's not really a male or a female mating type with Phytophtheras. They're really just mating types. And the individual that we have here is just one individual that has self-replicated millions of times. And the other mating type is not found here, as far as we know. And that's actually one of our really, really big concerns, is that if we were to introduce the other mating type here, would we get a recombination, and would it be able to affect other plant species than the ones that we already have now, which is a lot. So Phytophthora are not new to science, and you know, some very famous diseases have been caused by Phytophthora, like the Irish potato famine was caused by Phytophthora infestans. People who spend time in the Pacific Northwest might have heard of Port Orford cedar root rot disease, also caused by a Phytophthora. And then over in Australia, they have Jahar forests, and which are mainly eucalyptus trees, and but Phytophthora cinnamomi has affected many of those trees and caused a great amount of mortality. Consequently, Phytophthora cinnamomi is also a major agricultural pest. It affects specifically avocado industry. So, but ever since people started looking at Phytophthora ramorum, we are finding more and more different species of Phytophthora. We're finding species that we have never heard of before. And so we're kind of starting to get that baseline data that is so lacking in very many places. So this is a really old risk map of the United States, but I'd like to show it because it still is, has shown to be quite accurate. But also over the years, people have asked me, you know, like, wow, you know, how is it that you've managed to study a forest pathogen for 20 years and stayed employed? Because, you know, as a scientist, finding funding for the thing that you're interested in can oftentimes be very challenging. And when I look at this map, this is what provides confirmation to me as to why the government and other agencies have been extremely concerned about this introduced pathogen, because the potential is huge. And like I said, people understand that Phytophtheras are not to be messed with. They are to be taken very seriously. They can cause widespread damage. And so here in California, you can see all the way over on the left, you know, we're in a very tiny portion of the potential range of Phytophthora. And as you can see, like up in Northern California, up in Oregon, the climate is ideal, as well as the host composition to spread the disease is ideal. And so we haven't really seen a lot of disease up in the Pacific Northwest. It's really just starting to get going, but you can't ignore the southeastern part of the United States. And we do know that about 13 different states have had Phytophthora ramorum introduced through the nursery trade still, and have gotten out into different streams, but they don't seem to be causing widespread mortality like we see here in the United States. And we don't know if that's just a matter of time for more plants to be infected and to be spread. So there's a lot of people who are really concerned about this particular risk map and in the southeast of the United States. But the United States is not alone. They're over in Europe. They also have Phytophthora ramorum. And this is just a risk map that shows the suitable climate. This is not where Phytophthora ramorum is found right now. There's a lot of countries in Europe that have the pathogen, but really it hasn't done a tremendous amount of damage in the wildlands there. So what do we look for when we're going out looking to see if we find Phytophthora Worm Is. And first and foremost, the symptoms are really generic, meaning that there's lots of things that cause the same symptoms on these plants. And so you really have to sample and bring the sample back to the lab to know exactly what it is. It affects a huge host range of plants, but it doesn't necessarily kill everything. So just because it gets on a plant doesn't necessarily mean it's going to die from it. So there's really two separate diseases that Phytophthora Worm causes. There's the terminal canker disease, which is probably the one that most of us are familiar with because it causes the death of trees, and they're huge, and you can see them on the landscape. But there's a lot of non-terminal foliar hosts that don't really seem to die from the disease. So here's just a few examples of some of the foliar hosts. When you go out into our native forests and you look at all of the understory herbaceous plants, every single one of them is on the list. So it can get on them, but again, it doesn't really appear to be doing a lot of damage to them. Unless they're in a particular location where there's a lot of disease, and then you might see a few individuals die. But in general, we don't really consider this to be of great concern. And I think the big one here, people were always really concerned about redwoods. If you go to any redwood tree, I guarantee you're going to see these same symptoms, which is basically, see this little tiny black line on the tips of each of the leaves. You will see that everywhere. And we have sampled hundreds and hundreds of redwoods, and we very, very, very rarely ever are able to isolate it in the lab. It doesn't mean that that's not it, but sometimes isolating it from a sample is difficult. Tan oaks can get it on their leaves, but they can also be a terminal canker host. They're one of very few species that can be both. But here, when the pathogen arrives on a tan oak, it gets into the petiole. The petiole is where the leaf actually connects to the stem. And so this little area here is where the pathogen gets in, and then it moves up the mid vein of the tan oak and spreads out and causes a little bit of a canker here on the actual stem. So this is a sample that we've collected, and you can see the little sunken black canker. So that is likely the pathogen. I know it is because we sampled. I wouldn't show you a picture that isn't Phytophthora Worm for sure. But and then also here you can see it gets into the very tip. So kind of like the fleshier parts of the twig. But by far and large, the most important foliar host is California bay laurel. Bay laurel supports a tremendous amount of sporulation, which means it spreads it. So it is the most important driver of the disease is California bay laurel. And what you're looking for here on bay laurel is where you would imagine water to hang out. So as it rains, the water comes dripping down the bay, and as the rain passes, the water tends to hang out in these little tips. And sometimes if the leaf is hanging sideways, you would see, again, you'd see some dead part of the leaf on the side of the leaf. And so what you're looking for is this very jagged, defined black line, and then there's this yellow halo around it. And then you see some of these other little spots. But in general, you're looking for this jagged line and this halo around it. And that's the classic symptom of Phytophthora Worm Is. But again, you really would need to sample these and send them back to the lab to know for sure what is causing it. So some of the canker hosts, we have coast live oak, black oak, shreve oak, and canyon live oak. All can die from the pathogen. With enough inoculum load, it can get in there and kill these trees. But tan oak is by far the most susceptible host and the one that we're seeing the most mortality in across the state. And luckily, white oaks don't get sudden oak death. And I know that there's some beautiful white oaks on the preserve. And so that is one ray of hope out there. And so many people, I always get the question, why not white oaks? And that's a really, really good question. And I don't have the answer for it. I don't think anybody has ever really done sufficient research into understanding why white oaks don't get it. It's definitely a gap in the research. So what do the cankers look like on oak? So oaks in general have sap, which is red. It's a natural part of their physiology. The sap what makes it red is the tannins inside, and it protects the tree. So if a tree is naturally damaged, if the bark is damaged in some way, it will exude red sap. And so it'll look very similar to what you see here. But the major difference that we see with Phytophthora Worm Worm is that the bark is not damaged, but it is exuding sap. So a tree shouldn't just exude sap for no reason unless it was damaged by something. So that's what kind of keys us into to oak cankers. But again, you'd have to sample to understand, to know for sure what is causing the canker. Tan oak, again, one of the most susceptible tree species that we have here in California. Again, you're seeing sap coming out of the bark of the tree. And this particular picture shows a tan oak, and the bark is really smooth, so it's really easy to see the cankers coming out. But on that last picture, a lot of these trees have really deep fissures. You really have to get really close and look in between all the different grooves and stuff to see if you see any sap coming out. And then this picture in the upper right corner just shows, again, that the tip of the branch dying back, and you see this very clear line between the death and the green part, and that is a symptom of Phytophthora Worm Worm. So in Europe, they have different species of oaks than we do here, and so the southern red oak is very susceptible. Horse chestnut is also very susceptible, and as you can see, this really sad-looking European beech is oozing a bunch of bark, a bunch of sap. But one of the real big mysteries over in Europe is that they are seeing it in Japanese larch plantations. So for many, many decades, the UK has invested many hundreds of thousands of dollars to create and grow larch plantations for timber product. And so they also have Phytophthora Worm Worm. They actually have a different lineage of Phytophthora Worm Worm. They have the European lineage, and this particular European variety is a lot more virulent than the one that we have here in North America. Sounds, when I talk about this, it's interesting because it sounds like I'm talking about COVID. We talk about the European variant, and we talk about the one in Brazil, and so this is kind of like that. There's a different variant in the UK that is causing widespread mortality on a conifer. Here in the US, we don't really see mortality caused by Phytophthora Worm Worm on conifers, thank God. And this is something that we are very, very concerned about. And a lot of people in Northern California, where a lot of the timber happens, are very, very concerned that there is this potential that if we were to ever accidentally import the European variant of Phytophthora Worm Worm, and more specifically the different mating type of Phytophthora Worm Worm, that there could be recombination and that we could see mortality in our timber created species, or in any of the in any of the conifers like redwoods. And to be honest, so we actually we found the European variant just this year up in Del Norte County. It is the opposite mating type, and there was a massive eradication effort that was taken to try to eradicate this particular isolated outbreak of Phytophthora Worm Worm. This year, we are going to be looking at all of our samples for the European variant. Typically, you're just looking for whether or not it's there or not, but there's a new effort underway to see if we have more of the European variant here in California. It's something that we're very concerned about. So if you start going to the bases of trees looking for symptoms, you're probably going to see other things that are going on. And beetles are really common. The native beetles here typically only infect dead wood, but you'll see tiny little holes, and then you might see what we call frass, which basically looks just like sawdust. It's the tailings essentially of the beetles. So that's something that you would see. I mean, one thing that to know about when you see beetle frass or holes is that it indicates that there's dead wood there. So that's something to be aware about. And then you might see these charcoal balls. They're called Hypoxalon. Hypoxalon is actually an endophyte. So it lives within the tree for the tree's life, underneath the bark. And if something happens to the bark, whether a tree hits it or a car hits it or anything damages the bark to open it up to let oxygen in, the Hypoxalon can then come out and send out these fruiting bodies. So again, these fruiting bodies are oftentimes seen on trees that are affected by sudden oak death, but they by far do not mean that it is sudden oak death. It just means that there's dead wood underneath there. And the other pathogen that kind of follows along with sudden oak death is called armillaria, and some people might be familiar with honey mushrooms. But armillaria is a root pathogen. Sometimes it's pathogenic, and sometimes it's just a decay fungi. And so this is something that's really common. It's something that we're also quite concerned as there's more widespread mortality. There's essentially more food for armillaria, and armillaria can prevent recruitment of young trees. And so there's the potential for armillaria to cause lasting effects and creating canopy gaps into the future. And so this is kind of, armillaria would follow sudden oak death, and it's something that we actually are concerned of on many decades from now. So I talked about sudden oak death being in the United States, and it's also in Europe. This is just kind of a general overview map of where it is and what the different lineages look like. You can see the North America 1, so that's one mating type, and then the North America 2 is the other mating type. And then in red, you can see the European variety that has gone all over the place. And so this step here from Europe over into Northern California is the one that we've always been really concerned about, and it has happened this year. So we are very concerned about that. And I think I've already mentioned this. This is, but you really have to sample to know for sure that that what you're looking at actually is the pathogen. These are some really close-up images of some sporangia that happened on tan oaks. So tan oaks get it, but they can also spread it. There's sporulation that happens on the twigs. And then here you can see a really close-up view of symptomatic bays too. And here's a beautiful image of what it looks like once you plate it out and grow it in the lab. So I keep mentioning that it's really important to go and take these samples to the lab and then have them processed. And sometimes processing those samples can be kind of expensive if you just do it on your own. But every year, UC Berkeley puts on a sudden oak death blitz, and there's one hosted here on the preserve. I'm going to be hosting one as well in Carmel Valley, but they happen all around coastal California, from San Luis Obispo County all the way up into Del Norte County. And so you can go out and sample anywhere you want, including on your private property, and then bring the samples back to essentially you'd be bringing them back to Christy Wyckoff, and she would then send them to UC Berkeley, and you would get the samples processed for free. So this sort of information is super important. It gives you a very detailed image of where the disease is and where the disease isn't, which is really important when you're trying to figure out where you want to try to use your management dollars. But also, we're going to be they're going to be testing for the European lineage this year on every single sample. So it's really, really important to know where it is if we have more of it here in California. I think that you'll be seeing more information from Christy in the coming days on how to pick up samples and pick up packets. And so look for that coming up. So how is the pathogen spread? There is the human aspect, and I talked about the introduction in ornamental plants, and this is a really important pathway for not just Phytophthora worm, but for the next potential pathogen that may be introduced into our environment. But also people always worry about the potential to spread it as you're walking around on trails because it can be spread through soil. This is really a very rare event. From over the years, we've learned that really people walking around on trails don't usually pick up enough soil on their shoes and move the soil to a different location enough to actually infect another tree. So I think the guidance really is to just make sure you brush off enough of the dirt off of your shoes, especially when it's wet. But when it's really dry, it's so unlikely that you would be picking up big clods of mud onto your shoes. So in general, the best practices are don't move any plant material. Try not to move as much soil as possible. But really the main driver of the disease spread is in big wind-driven weather events. So in rainstorms with big wind, but not just any rain. It really needs to be a warm weather. So if we get some of this rain that we're really kind of hopeful for next week, that would probably going to be ideal conditions for the pathogen to spread, unfortunately, because we really need the water this year. But in general, these events are what cause major dispersal, major spread of the pathogen. So when I've looked back at the history of Phytophthora worm, we've really had about 5 major wet years, but we've also had 5 major dry years. And some of the information that's collected during the Blitz has provided some really key information to let us know where the pathogen is during the wet and dry years. And what they've found is actually that in the really wet years, they see the pathogen spread to all kinds of different locations. But in the really dry years, the pathogen contracts into these microclimates that are most hospitable for the pathogen, and actually goes away from some of the drier areas. And this is another really important tool when we're trying to make management decisions. When you kind of zoom in on the risk map, this was done by a different individual, but looking more specifically at California, you can see that, actually you can't even see Monterey County on here, but you can see that Northern California, Humboldt and Del Norte County have a huge amount of area and potential to lose a lot of trees. We are really just at the beginning of seeing the outbreak happening in Humboldt and Del Norte counties. And so we still have a lot to lose, even though we've been doing this and looking and doing research on sudden oak death for over 2 decades now. There's still so much more to lose. A recent publication by one of my colleagues has actually tried to come up with a number of the number of trees that we have lost. And we have lost at least 48 million stems due to the pathogen, but yet that was from data from 2012. And so since that time, there's been at least 150 million more. Well, there's still 1.8 billion trees that are at risk. So we're really, we can't really let down our guard between this number of 1.8 billion still remaining and the potential for the European variant to do even more damage or go into other species. We are remaining vigilant in the sudden oak death world. But there is hope. Not all coast live oaks have the same susceptibility. So some of them really have some immunity to the pathogen, but some are really susceptible. UC Berkeley has actually done a lot of work on the genetics, looking to try to see, well, is there something that is coded for resistance in these individuals? And from what I've heard, it's very complex. And as I mentioned, these trees don't have the ability to recognize this pathogen. And so one of our big hopes is that some of the new generations, some of the acorns that are produced from these trees that don't have the coding to understand that this is a pathogen. Some of the younger ones may grow up in a totally new environment with sudden oak death and basically be able to recognize it as they grow older. So this is something that we're really hopeful for. And in fact, we're not going to wait around for it, because especially with tan oak, that we don't see a lot of resistance. There's very, very, very little resistance in tan oak against Phytophthora worm. And so many years ago, UC Berkeley went around the state and collected thousands of acorns and had this massive seedling growing effort, and then subjected them to small amounts of the pathogen. Any of them that showed any sort of resistance whatsoever have been outplanted. And I know that there's some outplantings in Williams Canyon, and I know it burned in 2016, and I'm not really sure where they are right now, and if they survived or if they're maintaining them. But for many years, they did plant them out in Williams Canyon. This is a really important thing to take home. And if you remember anything from this talk, it's that that oaks don't infect other oaks. Oaks really need to have a lot of inoculum. They need to be infected over and over and over again to actually become infected. And so they typically need to be very, very close to a bay tree that is sporulating in really good conditions over and over again to become infected. There are individual cases of oaks that are able to, that get the pathogen, but without bays being nearby, but it's really rare. In general, most bays need to be near an oak to be infected, but oaks don't give it to another oak. So this is actually one of the best tools in our toolbox to try to fight the disease. So that brings me to what we can do. So you can monitor, and this is something that I'll talk about what we've done in this area with UC Davis. You can manipulate the stand, where you can't do it everywhere. So you really need to use every bit of information that you can get to make those decisions. Eradication efforts, I'm not really going to go into it too much, but eradication efforts have not been successful. Up in Oregon, they had very aggressive eradication efforts where they would clear cut 3 acres at a time and basically herbicide all the plants so nothing would grow back. And over time, the pathogen was able to infect the plants that were there. So eradication in general is not an option. You can try and use fungicides. You can try to, I think probably one of the most important things is to prevent human spread, but really you can start to restore some of these places, but education and trying to prevent the next pathogen, I think, is probably the most important thing that we can do on a larger, longer time scale. But for individual trees, if you have one individual tree or a handful of trees that you just cannot lose, you can treat it with this phosphonate. It's called Agrifos. And depending on how big the tree is, how much moss it has, you can use other things like pentabark. So you can either spray it on or you can inject it. It's not a silver bullet. You have to do it at least every other year. So it's pretty labor intensive over the years. It's not super expensive, but when you have that 1 or 2 or 6 trees around your house that provide shade that are absolutely important, and you can't remove a bay that's near it, you may want to consider using some sort of phosphonate. And I know that there have been trees treated on the preserve with phosphonates before. So this is something that you can get more information on the sodblitz.org website. And I'll show you that again. But so this is really on a small scale. These phosphonates can be effective at preventing the disease. So if you wait too long and your tree already gets infected, these phosphonates are not as effective. Sometimes they work, but really it's better as a preventative measure. But really, like I said, removing bays is going to be the best tool in the toolbox. So if you can keep bays away from the base of your oaks, you can protect them from ever getting infected. And I know that here on the preserve, that bays have been removed over the years, protecting individual oaks. It's super important. You know, I'm not a bay hater. I love bay trees. They are a native tree as well. We typically never recommend that people take out giant bays. If there's a giant bay near an oak and you want them both, there's things that you can do to thin out a bay so that there's more sunlight hitting the leaves, which the pathogen doesn't like because it really likes water and it likes the shady, moist conditions. But in general, we would not recommend taking out a really big bay. It's these little ones that hang out in the understory that are really close to your oak that are going to kill your oak. So it's really a choice that we have, whether or not you want to kill a bay to save an oak. So you can selectively remove bay laurels around super special oaks. And again, the sudden oak death blitz provides a very detailed map as to where the pathogen is on certain years and where it isn't in other years. And so if you can actually find the areas that are most hospitable for the pathogen and target them, then you may be able to actually drive your management dollars further. So in a year like this, if it's like a really dry year, you can go to those areas that are more hospitable to the pathogen and focus your management efforts there so that when hospitable conditions return, the pathogen would be less likely to be able to spread. And so how far away from your oaks do you need to remove bays? Well, it depends how big your oak is. The bigger the oak, the further the distance that you have to go. And here's that website, the www.sodblitz.org. There's a lot of different tutorials on there, lots more information about management techniques and directions. So I highly recommend you checking that out. This is from Matteo Garbalato's lab. So you can see here that if you have a small tree, you don't have to go as far away. So, but once you get into a bigger tree, you need to go a farther distance away. So, and when we talk about the distance, it's the drip line of the bay to the bole of the oak tree, and that's kind of our distance. And that distance is going to change depending upon the slope or prevailing winds can also should be taken into account as well. But really, none of us can really work at the scale at which is at a large scale, at a landscape-wide scale. But there is one thing that appears to be reducing the pathogen on this scale, which none of us can really imagine working at, and that is fire. And so this is kind of where I'm going to transition into some of the research that I've been involved with. And so I kind of wanted to just open it up for questions on some of like the background of Phytophthora and see if anybody had any questions about anything I've talked about so far.
Hi, Kerri, this is Carl. I saw on the map, it looks like the major infestation's up in that Del Norte area. I heard you saying, you know, wind carries it, but was there something else that got such an incredible concentration of it up there? You're talking about one of the least populated parts of the state, so I can't imagine they're doing a lot of ornamental planting up there.
Yeah, no, and I think that map that you're referring to, Carl, is a risk map. So it's only in a couple isolated areas in Del Norte County right now. So that risk map shows that if it was able to actually gain momentum, that's the area that is susceptible to being affected.
Yep. Mm-hmm. Yeah, you're right. Sorry about that.
No, but you know, even in very rural areas of Big Sur where there's no people, once the pathogen really gets established, then weather kind of takes over, and that's when it goes and spreads everywhere.
Frightening.
Yeah.
Thank you.
Yeah, you're welcome. Okay, well, if there's not too many other questions, then I'll just continue on. So I was talking about, you know, how do we reduce the pathogen, and what can fire do to do just that? And, you know, we all know that when fire comes through, it burns a lot of the foliage. So as you can imagine, if the pathogen is on the foliage and it kills it, it basically sets the pathogen back. But the real question is, for how long does it do that? And are there other interacting effects that we need to look at? So we actually kind of had that opportunity, and we still are having this opportunity to learn about this. So back in 2006 and 2007, UC Davis set up 280 plots in the Big Sur region. So up here in the preserve, where is the northernmost part of our plot network, and it goes all the way down to the San Luis Obispo County line. We had variable levels of mortality from this data. We saw that unfortunately, larger trees are dying faster. We're seeing lots more woody debris on the ground in areas that were affected by the disease. Maybe it seems obvious, but areas that had more bays were more likely to have the disease. And you know, every time we look at our data, we see more and more that bays. You know, bays are always involved in one way or the other. When we look for the disease, it's mainly because there's more bay trees. So we've been back a number of times to not all the plots, but subsets of them. And we've also seen a number of fires. So after 2007, we were thinking that maybe our work was done and we were going to move on as we were analyzing data in 2008. And then we had the Basin Complex fire. It was a lightning caused fire that burned about a third of our plots. And so we essentially had this natural born experiment to see how fire and the pathogen interact. But since that time, we've also had the fires in 2016, which I'm sure many of you remember quite well. So that was a human-caused fire, an abandoned campfire. And then now we've had the Dolan fire, another human-caused fire in 2020. So we've had very few plots left that have not burned. And so we've really kind of taken a turn to learning about the pathogen to learning about fire ecology simultaneously. So like I said, you know, how does fire affect the pathogen, but how has the pathogen actually affected fire severity across the landscape? So with the data that we have collected in these plots, I guess I should mention, so in these plots, we measure all the trees, we quantify all of the herbaceous understory, we're measuring all the dead fuel on the ground, and a whole bunch of other information in regards to slope and aspect and the different habitat types and canopy heights and stuff like that. So when we started to analyze the data back in 2008 after the fire, we quickly learned that fire is not an easy thing to study. It's very complex. It's very patchy. It's not homogeneous. There's some areas that burn really hot and some areas that don't, regardless of the disease. But the hope is with our data that we'd have enough of it to kind of iron those out. And so really, it's just important to understand that when we talk about how hot things burn, we talk about fire severity, but the fire severity is driven by many different things, including the amount of fuels, what the weather was doing at the time that the fire arrived in any one particular spot, the topography, and of course, the vegetation type. And so one of the first things that we found was that the pathogen is able to survive the fire. It mainly survived in areas that didn't burn very hot. It was also found in areas that had a lot of hosts. And in fact, it had a lot of bays. So the more bays that were left post-fire, the more likely that we're able to recover the pathogen immediately after the fire. There were a lot of plots, though, that we were unable to get the pathogen in that first year in 2009. But we also didn't have a very good winter that year. So once we had a better winter, we were able to recover the pathogen more and more. But these islands, so if you can see from this picture, there's this, a lot of these trees don't have any foliage on them, but this particular one very large bay tree had the pathogen. And so this actually represents a management opportunity where if you were to take out this one bay, could you potentially at least delay the reinfection of the pathogen into this environment, because you're taking out the one refuge of the pathogen in this post-fire area. This is not, it hasn't been tested yet. It's something that with the potential to do more prescribed fires in California that we might be able to test. And in fact, we have a couple of projects hoping to do something like that up in Humboldt County. So when we went out, we kind of expected everything to look like this. So this is a plot from up on Andromolera. There was a lot of disease in 2006. And after 2008, we were devastated. There were not very many trees left. But this is really one of the extreme cases of our plot. There was a lot of disease here, but it's also driven, likely driven by the topography. It's very steep there. So, but it's important to understand when we started to look at the data that it's not, sudden oak death is not a, it's a moving target, right? So that it changes over time. So here you see a healthy forest where there's deep shade. You have a tall canopy, the understory is open and the canopy is closed. So that would represent a healthy forest. Over time, as the pathogen comes in, it starts killing trees. And so you have all these standing dead trees with their leaves still on them. And those leaves have very, very low moisture content, which can lead to ladder fuels, which is when the fire comes up and it can bring the fire up into the crowns of trees, making a very dangerous situation for trying to fight the fire, but it also causes the surrounding area to burn extra hot. And so when you remove some of these trees too, there's more sunlight hitting the forest floor, so it gets warmer there too and drier. So at the intermediate stage, you have the trees that are starting to fragment, and you have a lot of fuel building up on the forest floor, and you have these standing dead snags, and you probably have some larger logs on the ground as well. And then the late stage of the disease, you just have it all. You've got tons of fuel on the floor, forest floor, and you have more trees continuing to die around you. Really, it's not a fun place to be. You have things that are starting to resprout a lot, and their resprouts are when the tree, the main stem of the tree dies, and it sends up little shoots from the base of the trees, and most of our trees in our area do that. So those resprouts then start to become affected, bringing dead fuel also close to the ground. So this wound up being really important when we started to look at analyze how hot some of our plots burned. So I'm going to show you an actual plot that represents exactly what I'm talking about. This is in Williams Canyon on the preserve. Many of you probably have hiked up that very lovely Redwood Canyon. So back in 2006, it was a closed canopy, shady forest. These are mostly tan oaks that you see here. Then the pathogen came in and caused a lot of damage there. So this was an early stages of the disease in 2009. You see a lot of dead leaves. You see some trees that are starting to fall to the ground. And you also see that more sunlight coming down into the forest floor. 2011 was really, really difficult. 2011, we took us 4 days to do this plot, which typically might take a half a day. It was really, really difficult to move around, huge amounts of woody debris on the ground. And so the later stages, you still see some of the snags or some of the trees standing they don't have their leaves or they don't have their branches, or they're on the ground. You can't really see them that much in this particular picture, but the understory vegetation is growing up. There's a lot more sunlight. And then the fire happened, the Sobrani's fire happened in 2016. And this was taken 6 months after the fire. You can see there's a huge canopy gap. There's not any trees remaining in this particular plot. Some are starting to resprout, but you can see it is a very, very severe burn area. So our data showed basically just that, is that the severity of the fire is related to the stage of the disease. I don't know what's going on with my slide here. It's a little messed up. But essentially, when you have that early stage of the disease, where you have the ladder fuel, you have the intermediate trees around them. The trees and the overstory suffer more damage. In that intermediate stage and late stage, you have the substrate or the soil burns hotter, and a lot of the trees are unable to survive such hot burns. So one of the other things that we found was that redwood, which is typically pretty tolerant of fire and doesn't seem to die from the disease, when you combine sudden oak death and fire, redwoods of a certain size, the intermediate size, were twice as likely to die. So when the fire comes through, it typically kills all the little trees. Little trees don't really do that well in fire, but the big trees, the big redwoods, they're fine. They can handle an amazing amount of fire. I'm constantly impressed by them. But it was that intermediate stage that we found this twofold increase in mortality that we are really concerned about. And as I mentioned, the soil burned hotter in areas that had the intermediate stage or late stage of fire, and that's predominantly due to the amount of fuel on the ground. As you can see from the picture on the bottom, all that fuel basically burned all of the nutrients out. They volatilized them into the air because it burned so hot. So we lost nitrogen and we lost carbon from the soil. in these severely burnt areas. But this actually brings up a lot of questions, because how does the nutrients then get back into the system? And so one of our current focuses is to look at the role of nitrogen fixing plants like Cyanothis. So Cyanothis is a bush. It comes back with vigor in the fire, after a fire. It's amazing. It's a nitrogen fixing plant. So here in the bottom left are these are nodules that are filled with symbiotic bacteria that fix nitrogen and re-input it back into the system. We don't really know how long these Cyanothis do that for. Nobody's really looked at our native species of Cyanothis to answer that question. We don't know how long it takes. So this is definitely a direction of our current research that I'm actually really excited about, because we spend a lot of time measuring Cyanothis, and it's really important part of the landscape, as much as people don't really like it because it's very difficult to move around in, but it is very important to re-input nutrients back into our system. So here's an example of a plot that came back into Cyanothis. So back in 2007, we had some disease here. This is in Big Sur, in the middle of Partington, actually. So post-fire, a lot of the trees didn't do that well. They're still standing though. There's a lot of woody debris. So this is just 2 months after the fire. We came back a year later, and you can start to see the understory vegetation coming back. You can start to see some resprouts coming out from the base of this tree. And we didn't really go back to this plot for a number of years. Actually, I think it was almost 10 years. And so I kind of thought it would be something like this. And when we got there in 2018, it was crazy. It was completely, so this is the center. There is a person in there. It was completely overrun in Cyanothis. And we basically spent 4 days measuring all the Cyanothis to see how many there were, how fast have they grown. And we are also collecting soil to try to get nutrient content, like how much nitrogen are they putting back. So I mentioned that a lot of the trees resprout. Almost all of our tree species here in our native ecosystems resprout. So they're designed, this is a fire adapted trait where they are designed to be able to be top killed. So here you can see these trees. The old stem has been burned, it has been killed by fire, and it actually, same thing happens with sudden oak death. Sudden oak death does not affect the roots of trees, it just affects the above ground. So it top kills the trees, and then they basically start their process of resprouting. They have these really cool underground structures called lignotubers, and these lignotubers are food reservoir essentially for the trees, and the bigger the tree, the bigger the reservoir. So some of these trees, we actually have no idea how old they are, and we have no idea how long they'll be able to resprout. Some of them may be able to resprout for very, very many years, and some actually have been resprouting for a lot of years. So one of the things that we notice in our affected plots after a fire is that there were fewer trees that were able to survive sudden oak death and fire long enough to be, or they didn't have enough resources to be able to resprout. So that meant that there was actually fewer trees that survived, but the ones that did survive actually were more vigorous resprouters than in areas that didn't have sudden oak death. So the fact that there's fewer trees, the fewer trees that are there have more resources then. They have more light, they have more access to nutrients, and they have more water because they're further spaced apart. So this potentially a good thing. You know, people talk about the forest being overstocked, so that we haven't had enough fire on the landscape in some areas, that there's too many trees, and that to build a resilient forest, you would have fewer trees so that they'd have more resources. And so we look at this as a positive thing. Not that we want lots of disease and fire, but it's something that we could say is a promising thing. And hopefully they'll be able to survive the next fire or the next round of disease. But our main concern really is that if another fire were to come in and then kill them again, how long can they continue to resprout? Do they have the resources to continue to do that? And unfortunately, we're getting the opportunity to learn about that because of the subsequent fires that we've had. Okay, so we have also learned here that that wildfire in general reduces the amount of hosts on the landscape. So when you look at the number of bays that are able, no, let me put it a different way. So when fire comes through, bays are really, really fire sensitive. So they don't do that. The above ground does not do very well in fire. Like I said, smaller trees don't necessarily survive very well, but in general, bays, no matter how big they are, don't do that well in fire. So when you go into the areas that have burned, you don't see as many bays in general. So fire has basically shaped a landscape in which that when there has been fire, you see fewer bays. So here you look at if you're looking at these graphs, if you're looking at the number of trees that have died over here on the left and the number of fires, the more fires you've had, the fewer trees that you find that have died from the disease. So again, and then if you're just looking at the amount of bay, you see fewer bay and when there's more fires. So this is, Christy mentioned that fire is very much part of my world. I don't think all fires are created equal. And so we do hope that we could get better fire on the ground, but that you need to understand that fire is a very important and integral part of our ecosystem here. And this is another way to look at it. This actually was a study that looks at the longer fire history. And again, it showed the same thing, that frequent fire dampens the disease dynamics by reducing the amount of hosts in the area across the landscape. So this is a hopeful part of our research that even though we've had multiple fires, it may actually be good for the disease. And so I'm going to just briefly touch on where we're heading with the UC Davis project. We are really concerned about the areas that have burned multiple times, especially in the area that burned in both 2008 and 2016. That's only 8 years between fire, and that we're worried that it caused some sort of immaturity risk, not allowing the trees to build up enough resources to be able to withstand the next fire. We definitely see that reburned areas experience greater mortality at the stand level, and that pre-fire disease history further exacerbates these effects. So when you have more fires or hotter fires, the trees are having a harder time surviving the next fire. So increased mortality in these areas may be explained by the increase in abundance of snags. So when the fire comes through, it doesn't consume all of the fuel. It kills some trees and then they're left standing dead. And so over time, as they start to fall, if there's not enough time given for them to decompose, they basically create a whole bunch of more fuel on the ground. So within that 8-year period of time, we're really concerned that it wasn't enough time for those logs to decompose, and essentially made for a more severe fire for the soil. So let's see here. So this is an example of that. There's this plot again on Andrew Malera, where it's had 2 fires, and now we have no trees left in the area, and not just in our plot, but in that whole area. There's just not a lot of trees left. We don't know over time if this is going to be if they're going to be recolonized by some trees, but this is definitely, again, this is an extreme case, but it is something that we are really concerned about. So as you can imagine, as a firefighter, trying to go out to create lines to protect people's homes and try to put out the fire, having this amount of fuel on the ground, whether it's from sudden oak death or whether it's from a very severely burned area, represents a huge challenge. You cannot put a line through the areas that look like this. And so maintaining fuel lines is extremely important before a fire comes. And you know, Governor Jerry Brown, before he left, say what you will about him, he did allocate a large amount of funding to do more management on the landscape. And the state is actively working at trying to eliminate the obstacles to getting more prescribed fire on the ground. And I'm hoping, you know, we're very hopeful with these 2 combined things that we will be able to make a change and make these forests more resilient in the face of disease and more frequent fires. And so, as you can see, I've represented a whole bunch of information from a lot of different people. So I'm just a small part of it. So I have to obviously say thank you to our funding sources. The National Science Foundation continues to support our research, the Forest Service and the Gordon and Betty Moore Foundation, and all these people. And of course, my boss at UC Davis Plant Pathology Lab. So with that, I will conclude and take any more questions. So thank you.
Thanks, Kerri. That was great.
Christy asked a question in the chat. Christy, do you want to ask yourself? Or I can ask it for you. It says,
I'll jump in here and see if
Okay.
anyone's got any questions for Kerri. It was an excellent talk. They get better every time she presents for us. This was a real incredible culmination of so much work, and really is telling the fire story much more clearly than the last time Kerri presented some of this research, when it was a little more uncertain of whether sod and fire were a positive relationship or negative for our overall forest health. So are there any questions? All of the material that she presented.
Hi, here's one. So I don't know if there's a percentage, or do we know about what percentage of the bay trees are infected with southern oak death? Instead of the prevalence of it?
Yeah, good question. You know, that fluctuates from year to year. So in some areas, in some years, at the extreme cases, 100% will be. And then in other areas, you might just find 1. So it really depends on the habitat. It depends on how dry of a year it is. And those are the 2 things that really are gonna make a difference. You know, some trees too they're not completely infected. So they may just have like one branch that's infected because it's in the shadier spot. You know, oftentimes you see them on the uphill side of trees. So when you're doing the sod blitz, always go all the way around the tree to look for symptoms, because the uphill side of the tree usually is shadier and closer to the ground and more moist, and so that's typically where you see the pathogen on the leaves. And so that's kind of what, like, if you really love your bay, but you know it has the potential to spread the disease, you could thin it and open it up so that more sunlight could actually hit the leaves and it'd be less likely for the pathogen to be there. Does that answer your question? It's kind of, there's no like fixed number.
Thank you. Yes.
All right. Any other questions?
Okay, and another one. There was this slide where you had an image of the Japanese, the Japanese, yes, exactly, that was spreading, and you can see that the surrounding forest was not getting it, only just like a patch. You know, are there any studies about like how this fragmentation is spreading the risk or reducing the infection rate between patches or a forest?
Good question. Well, I think in this particular picture, I think the reason that it looks patchy is that it's a plantation, right? So all of the planted trees are larch, and I don't know what the other things are. And so larch are the ones that are susceptible, and maybe the other species are not. But there has been research that shows that more contiguous forests are more susceptible. So that's something that that we're also looking at with our fire work, is that as the trees resprout and they create these big bushes, at first there's like big spaces between them. And so if one of them were to get infected, it would just stay there because it'd be hard for it to move. But once they start to get big and start touching each other and create this more contiguous forest, it's easier for the pathogen to move from one tree to the next, to the next, to the next. So these big, long distance dispersal events really only happen in like special conditions. So typically the spread happens on a more localized scale. So, so really contiguous, so fragmentation actually could help a little bit. And that was actually one of the eradication efforts that happened up in Humboldt County was we kind of nicknamed it the swath. So they basically had a 3-mile area, which was all timber company property, where they eliminated all the tan oaks in bays, and they were tan oaks that came back post timber harvest, poor timber harvest management essentially is what it was. But they were just clear cut all the tan oaks, thinking that it would create some sort of a barrier for the pathogen. It didn't work out. It didn't work out mainly because by the time they finished it, they found the pathogen on the other side of the swath, and that was unfortunate. But in general, I think that even the area that they cleared, I don't think was necessarily big enough for a wind-driven rain event to carry it. They say, I think that they've shown it's at least 1 kilometer or more in a big wind-driven rain event that they can see a giant dispersal event. Yeah, good question.
Thank you.
I've got a question. So, yeah. So you talked a lot about how there's so many different Phytophthora around, and we have some colleagues up in Portola Valley who had someone come out and look for Phytophthora on their property. And if I'm remembering correctly, they said they found 31 different Phytophthora. but they got very anxious because they were like, oh my God, there's so many different disease agents. But I have to presume most of those were native and not well understood. And so I'm just curious what we've learned about native species and potentially their roles in the ecosystem.
Oh, tough question, Christy. Basically, the verdict is out on a lot of different species, whether or not they're native. To determine a Phytophthora to be native, you need to have both mating types, and you need to see genetic diversity. And so that takes quite a bit of effort. And so I am not a Phytophthora expert, and I have been involved in heated discussions about other species of Phytophthora and whether or not they're native. There's a particular Phytophthora that's really widespread all around California. Whenever we look for Phytophthora worm, we typically find these other two species of Phytophthora. I've heard arguments between the experts, and there's not a consensus. Phytophthora are crazy. They're really interesting organisms. Even though we have both mating types of the North American, we have North American 1 and North American 2. But in the lab, when you put them together, they don't do anything. And when you look at the genetics of them, they basically have been separated for millions of years. And so, I mean, studying Phytophthora really begs you to revisit the question of what is a species. So are they different species if they don't mate together? I mean, typical biology basically would say, no, they're not the same species because they can't mate. So Phytophthora kind of have their own rules essentially. And I have heard a lot of these. There is a lot of new studies happening looking at Phytophthora. What do we have? What's that baseline? Like, what is normal? What is not? What are we concerned about? What aren't we concerned about? And nobody really knows. A lot of people have been looking at. So down in the Angeles forest, we have a colleague of ours who is looking at restoration areas. So restoration areas are a really big hotspot in Phytophthora research right now, because we continue to introduce Phytophthora through restoration efforts. It's so saddening because we're all trying to do the right thing, and yet we continue to struggle with introducing new Phytophthora. But are they causing any damage? Do they get out into the native environment? Sometimes, but are they causing damage? We really don't know. And so we have a PhD candidate who has some funding down in the Angeles forest to look at that. You know, are these native plants susceptible or not to any of these number of Phytophthora that you find out on the landscape? So it takes a big amount of effort to do all of these trials in the lab to have, oh, okay, I've got these 15 most common species and I've got 7 different Phytophthora. And so then you have to subject them each to tests to see, do we need to be concerned about it? So it's tough. It's really hard. It is kind of scary because at any point, any one of them could all of a sudden become the next Phytophthora worm worm. And there's no easy way, even at port of entry into the United States or to any country. How do we find the next Phytophthora worm worm? It's kind of the multi-million-dollar question. There's not a simple answer. I think some people would say the answer is to stop trade, and that's not going to happen. So what are the things that we can do? We can self-quarantine. That's been one of the things. I know the USDA is actually working on a nursery certification program, which I'm really excited about. I've been really excited about this for a while now. How do we get the consumer to demand certification, disease-free plants from nurseries? How do you, because like if I wanted to go to the nursery, I wouldn't know. I wouldn't know, like if I were to go and buy a plant, which I'm scared to do, you know, how do I know that they're following the best management practices so that you can buy disease-free plants? Right now you can't. I mean, you can look around, and if you know what the best management practices are, some nurseries are actually participate voluntarily in Phytophthora screenings, but not all do. And unfortunately, it's the small native plant nurseries that don't necessarily have the funding to follow all the best management practices, and therefore that's why we're getting this problem with restoration outplantings all over the country, really, with these different Phytophthora. It's really sad because we're trying to do the right thing.
So tough. Well, if there's no other questions, I want to thank everybody for sticking with us. I know we were over time, but it was just such a fascinating talk and so relevant to the ongoing conversations we have about land management and wildfire and what we can and cannot play a role in. I do want to invite all of you to join us at the Sudden Oak Death Blitz later this month. I will be reaching out to recruit volunteers. It's an excuse to conduct science while you're out hiking and maybe poke around a different part of the preserve than you're used to. So reach out to me, let me know if you would like to join. And I want to thank all of you for tuning in tonight. And most importantly, I want to thank Kerri for an excellent talk tonight. So thank you.
Thank you, guys.